Thank you all for tuning in today to this CellPress discussion. We're so excited to have you here today to talk about accessible high throughput experimentation. We'll refer to that as HTE throughout the event. And we're joined today by 4 leaders in the field.
For those of you that we haven't met, my name is Michelle. I'm a scientific editor at iScience, which is the interdisciplinary open access journal of CellPress.
And I'm Christina. I'm a scientific editor at Chem, which is CellPress's flagship chemistry journal.
So our plan for this time together is to engage in an informal discussion based on questions that were shared with the panelists beforehand and with the broad theme of making HTE accessible. So more accessible as a tool to enable researchers to access new data sets and new ways of thinking, independent of whether they work in industry or academia, or independent if they work on a small or a large scale. We hope that in this time we'll address myths about HTE, so things that you've heard that maybe aren't quite right, as well as give clear steps for the viewers to perhaps add HTE mindset or HTE to their own research. So we hope to give some concrete steps for you to take away.
Before we get started, if we could just take a moment and if you could introduce yourselves and give in a short 1-minute sort of speech what your role is now and how do you see yourself as a researcher engaging with high throughput experimentation. Maybe we could start with Jake.
Hi, my name is Jake Janey. I'm a scientific director at Bristol Myers Squibb. I actually run a technology group which has a number of components. High throughput experimentation is obviously a central component of what we do. And it's a unifying theme for many of our technologies. So we have biocatalysis, catalysis. We use HTE to explore material properties such as solubility and crystallinity, as well as using it to run design of experiment, et cetera. And I think the key thing that we've really tried to push over the years is marrying the HTE capabilities that we have with intelligence through data science, et cetera, to get better and faster and more targeted in our studies. So, as others might say, the technology has really evolved over the past 10 years, especially not only with how we handle data, but just the tools themselves are a lot more accessible and easier for people to get started. And I think the one thing that I've learned in my career is we've really undergone a lot of sort of changes over time where we had these big centralized fancy robotic workflows. And over time, the robots have gotten simpler and more purposefully built towards specific unit operations. And the software that people can use, as well as the tools themselves, have become much more accessible to many people. It really is kind of the default way of doing work nowadays to find the best conditions or develop a sustainable process. This is kind of the go-to way of working now and in the future.
Danny, do you want to go next?
Yeah, sure. Danny, I started at Merck in 2014, and I started off in process chemistry. And there I started to kind of dabble in HTE, and then it kind of led to a formal move into the high throughput experimentation group at Merck, where I really just fell in love with just the technology, how you could quickly impact programs. And I kind of leveraged the resources within the catalysis group. We had like large collections of ligands, of different types of catalysts, to start to engage with the academic community and start to, bring HTE into the academic realm through a variety of different collaborations. I'm now a director in a discovery process chemistry group. So still a little bit of process chemistry, but also, interfacing a lot more with discovery, and discovery chemistry moves fast. And so HTE is definitely, a really handy trick and tool to use within that space. And I've definitely, advocated for my group to continue to use this technique, kind of as Jake pointed to, not only for, screening traditional, synthetic organic chemistry, but also BioCat as well, which is a very emerging, technology or catalyst that we like to explore too.
Wonderful. Tim, do you want to go next?
Yeah, sure. Hi, I'm Tim Cernak. I'm an assistant professor at the University of Michigan and co-founder and CSO of the company Entes. And I have a long-standing interest in HTE. I've had the pleasure of working with, I think, everyone on the call in this space, or learning from each other. And I think that the community that has grown, as Jake articulated, over the past 10 years has been really exciting for us to watch. I think that a lot of things that we were excited about 10 years ago are coming to fruition now, and there's a lot of excitement in the space, enabled by accessibility of the tools, which I think is great, and I think a recognition that HTE is not about blind screening. I don't know if it was Danny or Jake just said that that we have very rational objectives. It's just about collecting your data in a systematic way and trying to cover a more broad space. And so I'm very excited about teaching the tools of HTE to young chemists, which is what I do in my academic role. Very excited to have this discussion today.
And last but not least, yeah.
Hi, my name is Matthew Gaunt. I'm the 1702 Yusuf Hamid Professor of Chemistry at Cambridge in the UK. I'm a synthetic organic chemist, and broadly speaking, my research interests span the development of new reactions, new reactivity concepts, enabled by catalysis through to biomacromolecule functionalization. And over the last 5 years, we've established almost a third of my group now is active in this area of high throughput synthesis. We've created platforms based on initial collaborations, in particular with Merck and also others subsequently. And within this area, my specific interests really are trying to build platforms which enable us to discover and develop new reactions, as well as generating data or collecting the data from these high throughput experimentation platforms so that we can work towards an element of predictive chemistry in the reactions that both we develop and also that could be more broadly used. On top of this, and this is a result of some of the initial work that went into this area within my group, I'm the director of what's called a Center of Doctoral Training, which is essentially a PhD training program, graduate training program, which hosts up to 60 PhD students in this area of what we call next generation synthesis, of which high throughput technologies and data-driven science are the major component of that. And this, alongside other initiatives in Cambridge, we're trying to build essentially a hub for this area of science that has a sort of an outward-facing objective to work with both academics nationally and internationally, as well as industry from a SME perspective, all the way up to the global partners, which we've enjoyed many fruitful collaborations with over the last couple of years.
Thank you so much for those introductions. And now that we all kind of know where we're coming from and the diverse backgrounds that our panel is coming from, I think we really just want to start with what are the biggest myths associated with HTE? I think, you can see a lot out there, what people have misconceptions about HTE. And alongside that, what are the most underrated capabilities that maybe people don't see when they hear HTE?
I can maybe just start here because I think I want to pick up on something that Tim just mentioned. And I think the biggest myth in HTE is that it's mindless screening or at worst combinatorial chemistry. This is complete fiction. Actually, I would argue to use HTE to its full capacity, you need to almost think more about the chemistry that you're doing and not less about the chemistry that you're doing. And so I think trying to get that message across to the community is absolutely fundamental. There's a lot of skeptics out there, right? Naturally, when something new comes along, people think it's dumbing down. Chemists don't have to think anymore, but I would say it's absolutely the opposite. It's you really have to think about what you're doing here, much more so than when you're doing conventional batch style chemistry.
Yeah, I think that's a really good point, Matt. The idea that you're throwing a bunch of stuff against the wall and hoping for a result is very much not the case. In fact, the one question that we always ask people who are first embarking in that endeavor is, what's your design philosophy? How are you going to explore that space? And you can get very fancy, as we have with Bayesian optimizers and, in approaches like that. But there's also really simple statistical techniques for exploring that space. But fundamentally, you have to have a hypothesis and you have to have a way of exploring a very wide space. I'll give you an example. We were recently running just a simple Mitsunobu reaction, which when we looked at the variables is 180,000 configurations. There's no way that we'd be value added to test all 180,000 for, just a Mitsunobu reaction. But, by applying a really innovative design philosophy that we explored with the Doyle group and the Adams group at Princeton, we're able to find perfect conditions, 3 perfect conditions with only 50 or 40 experiments. That's like 0.02% of the possible combinations. So I think we're going to see more of that in the coming years. We've gone from people trying everything to DOE, and now to these much more advanced, learning techniques that are really fundamentally changing the efficiency with which we can explore that using HTE.
If I could add to that, as someone who started off in process chemistry and, wasn't a believer at first, because when you do HTE, everything is micro scale, and you start to wonder, I'm doing this on milligram quantities. Can I really do this on a kilo? Is it really going to scale? And I'm here to tell you that it does. And, I've seen it time and time again. And so I think that's another myth, is that, what you observe in, a 1 mil vial on a 1 mg scale will never be able to be like a real reaction that you can actually develop, is a total myth. And it's been busted a bunch of times.
And I'm so stoked that you brought that up, because I think that's one that, we always get in this space. I think maybe a myth that I would put out there for younger students, and kind of piggybacking on the earlier thread, is that you run a lot of reactions and you'll get your result. Or I think when I was a grad student, we would count our reactions, right, and be like, oh, I ran 5 reactions yesterday, so I did better than my lab mates. And as I was starting a group that was going to focus on HTE, this really weighed on me, because I don't want students coming to me and be like, oh, I ran 96 reactions yesterday, so I did great. I mean, you can easily run 96 failing reactions, and that is not a very good use of your time. You can easily run 1,500 reactions that fail. And so that's just not a very good use of time. And so I think that, the feeling that running more reactions will necessarily get you there, I think without thinking is probably quite flawed. And so, as Jake pointed out, right, I mean, for a very specific transformation, you could have 180,000 combinations, I think was maybe the number. So, whether you run 1 reaction or 96 reactions in that space, you really need to think hard about which 96 reactions you're going to pick.
Thank you, guys. I think these are some really interesting points, especially as regards the scale of reactions. And I hope we can touch on that more later. Just to mention something that has also come up, automation. Tim, you just mentioned that, running more reactions, which naturally you could do if automation is not necessarily the best target. What do you see as the role of automation across the fields of HTE? Thinking about this in terms of chemical synthesis, but also across the life and pharmaceutical sciences.
Yeah, I mean, if you guys don't mind, I'll start there. I think the beauty of automating these processes is that you get very systematic data capture, and so I think that's. I mean, we've talked about data and the intelligence aspect of this, and so having systematically captured data, I think, is really a beautiful thing. I think it's also no secret that a lot of what our chemistry community has done has been stealing tools from biochemistry automation. That has been well established. I mean, I love to point out that the 96 well plate was invented in 1950. And so, biochemists have enjoyed an evolution of infrastructure and community around the well plate as an experimental tool. And so synthetic chemists, I think, have maybe, rightfully had to in the early days compromise, because well plates are not really the way that we would want to, it's not great for organic solvents, and there's air sensitivity and stirring and all that. So I think earlier practitioners had to maybe compromise on some of the experimental capabilities, and that's still true when you automate a process. But I think that automation, can increase throughput, but it's still, it requires, I think, a lot of effort and thought to pull that off.
Yeah, Tim, that's a really important point. The thing that people forget, they all think about productivity, efficiency, blah, blah, blah. It's all about data quality. I always say, would I rather work with a data set that is 100 experiments run by 100 different chemists, or, a 96 well plate or 100 run by a robot? The data quality and the variance is going to be much lower, because even if it's absolutely incorrect, i.e., there's some fundamental way that you're running the experiment that shifts your results a bit, at least it's an internally consistent high fidelity data set that you're getting out in the back end. And for our purposes, especially in an exploratory sense, that is of critical importance if we want to actually learn something from that particular transformation.
I think there's another side to automation as well, which is really important. And, not all chemistry is automatable, if that's the right word. But some of it is. And that, at the end of the day, if you can get a machine to do the boring stuff, it means that you can put your mind to the more interesting stuff. I mean, I think of an example of, if you wanted to manually weigh out 0.5 mgs of palladium acetate across a 96 volt plate, that's mind-numbingly boring. But if you can get a machine to do that for you accurately, then I'm all for that. And it means that, I was going to say myself, my students at least can perhaps think about how to use their time more productively. And I think the challenge in automation is actually getting the full range of chemistry to work on automated platforms, because certainly from our experience, it doesn't. I mean, it's a lot of it is an engineering problem to get chemistry to work on different scales. And so I think, even within the area of automation, there's a ton of intellectual challenges that still need to be solved. That if we want, we're light years away from fully automated chemistry, but it's still a goal that we should be looking for, because we can save so much time. And as has been pointed out, the data quality of automated the data set generated by automation is certainly going to be way more reliable in terms of the bigger picture for data-driven science.
I think you save a lot of time, but you also can acquire more data. And maybe that's a good thing or a bad thing, but as when you go home, you could potentially keep using automation to sample your reaction plate, get more time points, get some kinetics, and then the next day you have like a beautiful profile that you can then study and understand more, and you're not there over your plate constantly taking time points. And so with that regard, I think it's also a benefit. And I also wonder if the pandemic kind of accelerated automation, as far as like people, would just set up their robots and then they go home, and then they could, be acquiring data and, not have to be in the lab.
Yeah.
So go on, John.
Yeah, I was going to say that divorcing the execution from the design and analysis is a real future direction that we're seeing. There are already companies with cloud-based labs that have essentially offloaded the execution component to those very automated processes. And I think we're going to see more and more of that over time. Chemists will become increasingly, quote, data scientists, thinking about hypotheses and how to test them and how to interpret the data. And a lot less of the execution component will be done in-house or by their own hands. And I think that's an important point that the pandemic certainly has highlighted, like, hey, we can do a lot without actually being in the lab if you have the right, relationships with companies or with the right capabilities internally.
I think those points. Oh, do you want to add something, Matt?
Oh, no, it's fine. No, so I can talk about this later because it's going to come up later anyway.
Yeah, I think this really leads naturally to what we wanted to talk about next. Is that one of the cornerstones that you've all brought up here is that you need to have the ability to handle bigger data sets and analyze more data. And you kind of even make the suggestion that chemists will become data scientists, sort of, in how they approach the data that they're collecting. And how do you think the role of data analytics, obviously it's a huge role in what you do, but also data visualization in your day-to-day work? And how do you see this sort of progressing? I think we've touched on it, but maybe the data visualization we haven't touched on yet.
Yeah, I'd like to start on that one, because the thing that's, I think, the quiet revolution that's occurred in the past 5 years in particular is just the number of amazing open source solutions for data visualization, et cetera. I mean, the folks in my group are, super proficient in using R. That's one of their favorites. They use Markdown reports. They have off-the-shelf visualizations in Python, et cetera. There's Tableau, there's Spotfire. You actually have almost too many great tools to choose from in some ways. But that's not really the choke point. It's always about the creating the tidy data set. In other words, integrating the different sources that we have in the lab, whether it's an analytical measurement or it's the actual execution from the robot. That's where the people have to spend their time. It's not really the visualization. Once you actually have the data set in a format that's consumable by these tools, then it's pretty trivial to express that in whatever format is useful to a team to understand what's going on.
I'll jump in because I just love this topic, and I love data visualization. And, I think that as a PhD chemist, we were always so envious of the cell biologists, right? That could make like the glowing cell images. And I think that now the organic chemists through these big data regimes and data visualization have the opportunity to really make some beautiful images, and I think that's fun. But I think that there's also some. I mean, we have had many opportunities to use that to make discoveries. I think that like the number of times where I have looked at a kind of big scatter of data, and then you end up grouping the data in such a way that, it kind of looks like a buckshot of in the beginning of, we use yellow and blue dots, but then they cluster in ways as you find them. And we've actually uncovered mechanisms of reactions that were completely unknown to us just because we group the data just kind of playing around and a pattern pops out. And I think that's really, to me, what data visualization is largely about, is looking for patterns. And so I think that, there are patterns that emerge that couldn't be seen other ways, but there is also some beauty, I think, in the data visualizations that come out.
And one of the things that, my group doesn't particularly deal so much with the data visualization, because we made a decision that I'd rather leave that to the experts as opposed to try and, I don't believe that a synthetic chemist is going to be a data scientist, nor do I believe a data scientist is ever going to be a synthetic chemist. And so we've worked with experts in data science rather than, to try and do it ourselves. Because I think, you use the biology analogy. It takes like 8 years to train as a cell biologist. I'm not suddenly going to be a cell biologist overnight. I won't become a data scientist overnight. But I think what I think is really important is that chemists are at the table when it comes to the data revolution in chemistry, because what we don't want to do is end up handing this over to an area of science that doesn't really understand the challenges involved. And I think we can shape this. It's a really interesting stage because there's actually not very much data available, good data available in synthetic chemistry. We have a real chance to shape this going forward. But we don't want to just give it away. I think it's quite important that we're involved in that conversation. Even if we're not the experts, we can certainly help to shape how the field uses the data that we can produce.
Yeah, great. I think this topic of clean data, of acquiring large data sets is really an interesting one, especially for the field going forward. And I think it touches on this idea that there's a gap between HTE use in academia and industry, potentially. I know several of you have worked in both. Especially when you start talking about, having a specific data scientist to look at your data, or having your students acquire those skills that they need to work on this scale. What do you think are some of the biggest barriers that might be perceived or real to move into HTE on the side of academia?
Cost. This is not cheap science. And maybe Tim can also back me up on this. Tim obviously came from an industry path in the first instance, and so saw the other side of the fence, where I'm not suggesting that everything grows on trees at Merck, but there still has to be budgets put in place. But from an academic perspective, it's quite hard to get started because there is an initial quite big financial outlay to buy. You can set up various forms of high throughput experimentation. 24 reactions is still high throughput compared to one. It's all relative. But if you want to invest in something that does 1,536 reactions, that's an investment of, $100,000. You need a mass spec to attach to that, another $100,000. So already you're starting to add up. And, we're very lucky that we've been able to find ways to purchase equipment in this area. But it's not easy to start out in academia in this. And so I think a lot of people are put off by this initial buy-in, which is often quite difficult to overcome.
I would somewhat disagree with that, just because I've been an advocate that HTE is accessible. And to your point, 24 wells is still HTE. And that is something that a student is very easily can grasp, set up, their parameters, and they can assay by NMR, right? If they want to get the data. And so I've been a proponent of just trying to get folks in that HTE mindset. And I agree, 1536 may never be a realization for an academic lab, if not maybe just a few. But I think getting them more familiar with the 24, 48, 96, I think is totally graspable, and all it requires is simply a glove box, an automatic pipette, and you're kind of like good to go with that regard. And I have a few examples that we can get into later, but I've definitely seen, HTE becoming, something that students now want to do and that they see as a potential to help them in their projects.
Yeah. If you don't mind, if I can just jump in so I worked at Merck with Danny and Jake, which I don't think I said in my intro, but I had no idea what a 1536 experiment cost in those days. And I mentioned earlier that it's easy for students to, or I shouldn't say it's easy because it hasn't yet happened in our lab, that they've run 1536 failed experiments. But that is my biggest fear because I now know it cost me around $500 per experiment in consumables, just in the well plates and tips. And so I had no idea what that cost, when I was in industry, and I do now. And so I think that's kind of one of our markers in our lab is like, don't burn through $500 and get unusable data. For instance, if you use like wet solvent or something, right, that just kind of threw out all the data. But I also totally agree with Danny that I think that it's a very accessible thing. And I also think that the pandemic has probably launched a wave of publications that we've not yet seen, where I think probably a lot of people, a lot of organic chemists learned how to code over the past year, and there are more increasingly data sets becoming available in the literature for people to work with. And so Matt, I totally agree earlier that I think that this field is launching a lot of conversations between data scientists and organic chemists, and so I think it is very much a collaboration. Back to this data visualization aspect, but I do think a lot of organic chemists have probably taught themselves how to code and to facilitate that conversation.
Yeah, I was going to agree with Danny and Tim in the sense that I think it depends on the kind of throughput you're talking about and the types of experiments you're looking at. If you can run a 24 well plate and get to your answer, that's likely good enough. And certainly for some transformations that we actually have a lot of data on and understand, the number of experiments that you have to do is going to decrease precipitously. For CM couplings, I don't think there's a big driver to have to run a 96 well plate for that, right? You could probably, deploy a predictive tool and it'll suggest like 5 reactions that you should run and then another 5 or something like that. So this kind of iterative parallel experimentation in some ways is a lot more efficient to explore that space. And it doesn't create a lot of burden on the experimenter to set up and have all the consumables and automation at their disposal.
I think it's important to point out depends what the goal is, right? So a 24 reaction data set is not going to solve you any predictivity problems, right? You need 1,000, you need 2,000, you need 5,000. And I think it's all, this is, I think, the great thing about HT, and it's, as Danny said, it's all relative to the need, right? So 24 reactions is still high throughput compared to 1 reaction. And I think, that's obviously, accessible within a lab, but if you want to develop a 3,600 data point set, mm-hmm. And actually, there are people that have done that manually, but that is a gargantuan piece of work if you don't have automation equipment to be able to do that. And so I think, it's multifaceted, right? This whole field, and the HTE. Actually, I wonder if we need to add an extra constant like UHTE or something like that, ultra high throughput. Maybe there's an extra dimension to this, but I think the key. And just to emphasize again what Danny said, right? The idea is getting people thinking about chemistry in this way there's some skill sets in there, and there's also an activation barrier to get people to think about it. And I think, in academia, I think what we can do is train our students to think like this, because actually I think in industry, I think it's a little different. I think, because these platforms exist, there's a mindset already in place that, this is expected almost. Whereas I think in academia, it's not necessarily the expectation. And we have to change that. We're starting to change that, but it takes some time. And it comes back to this myth a little bit. HT is like dumbing down. Actually, it's, I think, completely the opposite. Even, on 1,500 reactions, there's no dumbing down in that. So it's how to get the message across, I think, but it's accessible in whatever context you want to use it.
Yeah, I really like this discussion. I was just in grad school a few years ago, and I never even thought to run my catalytic reactions, multiple of them, concurrently at the same time. So just as adapting this mindset, if I would have known a little bit more then, maybe I could have applied it. And I think that's a really good point. But also to see what you actually want to get out of it is of course. Obviously a major part of all of this. I kind of want to switch gears a little bit because Tim, you said something that I thought was interesting about, how much the consumables cost. But also I think from a sustainability point, HTE is kind of touted as a big step in sustainability and especially green chemistry, removing some hazards from the lab. I kind of want to hear, is this a myth or is this definitely what you feel as members of the field? This is what it is doing for the field, or I kind of want to hear some of your perspectives on this.
Well, I do think to the point of consumables, right? It's not just a cost. There is an environmental impact of having like a lot of disposable tips and vials, et cetera. But if you're talking about actual material usage, for example, and solvent waste and everything else that would go into, running an HT where you can miniaturize and number up, I do think that there is a pretty tremendous efficiency gain and a less environmental impact overall. Although, I think it's a worthwhile study for someone to actually do that type of kind of life cycle assessment and actually look at that problem. But I do think, just solvent waste alone, you can make a very compelling argument that it's still more efficient to operate in that manner, despite the fact that you are using some consumables along the way.
So on the industrial aspect of it, I would say that, maybe there is some, consumables that you use, but if you're screening for, say, the utilization of greener solvents or base metals to replace a transition metal, I think, when you start to think about scaling the chemistry, that we do to manufacture API, that's green chemistry. Like that's where it counts. And so getting that in through HTE, awesome.
Yeah, I mean, it's not just about yield. And I know, I'm sure Danny and I, when we talk to academics, we always have to impress upon the fact that, yeah, yields are great, but we care about, what was the solvent? What was the concentration? What's the volumetric productivity of this process? These are all different hypotheses that you can generate information on through HTE. So we're typically talking about multi-objective optimizations. That's kind of our default process chemist thinking. Most of the time, it's not just about making the material, it's making a material that meets those criteria for the business that are cost effective and green. So, I do think to Danny's point, if that output gets realized on scale, you do have a tremendous value because you might have discovered conditions that you might not have otherwise. And that translates to, savings in terms of waste.
I think you said as well that, with CN coupling, I think you're right that there's these beautiful predictive models that have become available because of HTE, I would say, and running 5 reactions now instead of previously, I don't know, maybe thousands somewhat manually would have been status quo 5 years ago. I'm not sure. But I think that the efficiency of information extraction has improved a lot from it, and that's inherently green.
I think from an academic perspective, it's possibly a little bit different because I think the sustainability angle, especially in process research, obviously it outweighs anything that we would do in academia. I'm not sure if there's apart from what HTE can potentially deliver through discovery or optimization of a reaction in academia, I think that the immediate benefits are probably a little less. I think to offer a caveat to the whole argument, I mean, if you want to run my ultra high throughput experimentation, it doesn't cost me any less, or I don't use any less material in the overall scheme of things because nobody sells 2 milligrams of a various amine. I still have to buy 25 grams of it, of which I'm probably never going to use it again. So I think that there's a few different ways of thinking about this, in particular in terms of data generation data set generation and reaction development in academia. But I think, holistically, if you take it can investment in this at the early stage will reap the benefits further down the line when you've identified the base metal or the reaction in the solvent, which you never thought of trying, but is much more environmentally. But those are obviously, unsurmountable benefits. But I think academia, it's like a bit more of a tool to speed things up rather than we worry too much about this. I probably shouldn't say this, but we don't worry too much about the sustainability angle just yet.
But Matt, to your point, right, there are some long-term, you're playing the long game, essentially, right? We're looking towards academia to come up with the next Suzuki reaction, right? We want them to come up with a transformation that allows us to cut 3, 4, 5 steps out of our route. So, as these technologies proceed, if Matt's able to find a brand new bond disconnection that's going to have a much wider impact across the entire industry if it really allows for simplification.
Matt, I think highlighted one of the major pain points for me is vendor availability of reagents. That I think HTE requires a broad diversity of reagents in very small amounts, and that's not really I think that's a pretty tricky commercialization model, right? That the industry, the commercial vendor of chemicals industry is based on selling as much, the biggest bottle of something that you can, right? And we're asking for 1,500 things in 2 milligram amounts. That's a gap that I really hope can be overcome in the.
Yeah, ironically, I reckon the vendors will end up charging us more for less. It's like biological reagent, right?
Right.
Yeah, I think these are all really great points, and some things that I never considered at all as a little bit of an outsider to this. So thank you so much for bringing this up. One thing that came about is kind of how does anybody watching, how do they adapt an HTE mindset, or how do they even think along these lines, especially when it wasn't ingrained in them at all? It wasn't ingrained in them in grad school. It wasn't ingrained in a postdoc or a position. They have no idea. They're coming very fresh. They see all these exciting papers, all of your papers out there. They see them and they want to do it. And along those lines, there's also a very active online community as part of HTE. We recently saw a curated list of recommended papers from Nessa Carson, who's like a huge figure in this field, and I wanted to give her a name drop. So what other resources do you kind of want to add if somebody was to come up to you and see your paper and be like, I want to just do something HTE in my own lab? What kind of resources would you point them to? Would it be your own papers? Would it be other people's papers, blogs, courses, company resources? I don't really know what it would be, but I'm just curious.
So I think Nessa's list is a nice start. I think she does have some good references from companies such as Merck and others who have kind of put together a beginner's guide to HTE. The steps themselves are pretty simple, but I think the part that you just alluded to is how do you get people to think of that sort of hypothesis testing mindset in parallel? I think most, myself included in our training and as a PhD student, always are thinking of one factor at a time. In fact, I was specifically instructed by my PI at the time is I should only change one variable at a time, which is completely against what you learn as a process chemist, right? You care about interactions. You care about what happens when I toggle these 2, 3 switches at the same time. So I do think that there's a little bit of almost like not only the how-to part of it, but also that design mindset that actually needs a little bit of grounding in fundamental statistics of one factor at a time optimization versus, traditional off-the-shelf DOE factorial type of optimizations.
The one angle that I've taken, which is, I guess, my privilege of working at Merck is, engaging in academic industrial collaborations with a variety of different institutions and different PIs. So whether it be, St. Olaf, which is an undergraduate institution, or the University of Michigan, just, showing them the technique and how, we found best to do that is you just invite the students to Merck and you just give hands-on training. You equip them, you teach them the skill sets, and then you send them home with like a block that they can do themselves, or you send them reagents. And there's actually a lot of, a few companies now are starting to kind of like produce like the 24 well set of like, say, CN catalyst or peptide coupling reagents and whatnot. But, I think what's really awesome, and that's, you only have to change one mind, and hopefully, they have the courage and influence to start to change others as well. And so, for instance, I recently completed a collaboration with Corinna Schindler at the University of Michigan, and had a couple of students come out and visit, showed them the equipment that we have. And to Matt's point, HTE can be expensive, and they thought maybe it's best to build a HTE center at the university for other groups to use. And so, by leveraging their connection with us, we were able to show them what to actually buy, not to, buy into getting robotics, because they don't need that. And now it's like a facility that students can just go up to and use. And that was all the result of just having 2 students come and visit and do some stuff on their own with me.
I mean, I think that's crucial to the future anyway of this field, is that you find a way to disseminate the practicalities of it. So, and what you don't want is in academia, every single lab having their own individual HTE platform, because it's just the cost is going to be astronomical across the whole field. You want to build centers, you want to build outward facing centers where people can come in and learn about it or use it, because not everybody wants to bring it all in house, especially on the higher end, higher throughput end. So I think that expertise, and it's great, Merck have been fantastic at this over the 5 or 6 years that we've worked in this area, have been very open about how they do the science, and they've always been very helpful when people have had questions. And I think that's key to this going forward, because there's a ton of expertise in industry here, actually much more than there is in academia.
Yep.
And I think in the longer term, actually, you could argue that there's industry should invest in this in academia, because actually the people, you're going to recruit these people in 5 years' time, and it's much better if these people are trained in this area through their graduate studies than have to train them yourselves, or at least train them again. So I think there's a synergy here, which I think is really good, and having people come in, like you said, they do is really fantastic. And that will spread this field, I think.
Yeah, I hope that there are more shared facilities like that at different universities. You see that with NMRs, right? You don't have NMRs for each group unless you're dealing with something explosive. Like when I was an undergrad, Phil Eaton had his own because it was likely to blow up. But nonetheless, right, you have these shared facilities, and the funding agencies should recognize that, hey, this is the future, and having these 4 different universities and institutions is really crucial. And then partnering within industry, right? This is all kind of pre-competitive. We are an open door when it comes to the methodologies and the techniques. You can talk to any of us, and I'm sure that anyone would be happy to kind of chime in and give their own, advice to any university or institution that wants to get started on this endeavor.
So on behalf of the University of Michigan, I want to say thank you to Merck for all that you do for us, and to BMS too. But I want to answer the question a little bit differently in terms of, what can a new user do? If there's a first-year grad student out there or a second-year grad student listening to this, I think, we talked about thinking in this HTE and you read the literature, you take a paper, and then you go to your fume hood and you try that, how you would riff on that paper to make your new discovery work, right? Like maybe you take their catalyst and use it in your particular setting. Try doing that with 8 papers and just see if you can, on paper, just draw an array of 24 reactions that would kind of line up the solvents the right way, so you could do it all at the same time. I think as Matt pointed out earlier, I think it's a really it's a really thoughtful process to pull that out. And it's a lot of fun, really, to kind of play this Rubik's Cube of reaction conditions and try and build it into your array. So with no equipment at all and no network of people that that you can go and do this, I think you can pull 8 papers from the literature and try the paper exercise and bring it to your PI and see what he or she says.
I'm actually a really big fan of digging through the supporting infos, because in there is a lot of optimization, and you can see all of the variables, and you're those are the 4 bases that I want to look at. Those are the 8 solvents. Those are the catalysts. These are the loadings, and it's really rich. Usually I don't even read a paper. I'll just go straight to the SI and just start to dig around.
Well, thank you all. This has been really great. And I think there's some really good advice in there for our audience as to how to get started. I want to be cognizant that we're coming to the end of our time. And so for our final question, I want to ask something a bit fun. And that's, is there something in the HTE community today that people would have seen as science fiction if they had heard about it a few decades ago? And looking forward, what do you think the next advances like that, the science fiction, I know Jake Janey is a big fan, will be?
Well, I guess I'll start. I think a lot of these kind of human out of the loop optimizations, I think, if you told me 5-10 years ago that you could actually build a robot that has an optimization algorithm directly piped in. Now, this is particularly applicable to flow, but, you could definitely envision, high throughput robots that are doing this. And we've seen some really nice examples already. I would have definitely said, you can't do that. Chemistry is too complex. But already for, some pretty standard transformations like amidation, Suzuki CN, the bread and butter of chemistry, those things are becoming increasingly, automated to the extent that you don't need people in some cases, other than to say, this is what I want to do. So that to me is really cool. I geek out about that, obviously, but I think that's where we're definitely going to see more and more of that in the future.
I can go next. So I guess one of the reasons why I got into this area was when you think about molecular biology and advances that have been made there, I mean, just the ability to decode the human genome is like a staggering achievement in science. But why can't chemists or synthetic chemists, and I'm going to coin this phrase, which I've been dying to use for years, we've never had the opportunity, why can't we decode the chemical reactome or something along those lines? We should be able to program synthesis in a way that, we understand what it all means. And, this is a long term. I don't know when we're going to solve this, but to be able to do the same that geneticists can do now, but with synthesis, and then, think about what are the layers below the genetic code, the epigenetic code, the epitranscriptomic code. This is all there in synthesis as well. And so being able to understand, predict, and then harness that will open up the opportunities for us, I think, to try and think about problems which we just can't even comprehend yet in making molecules. And I think that's what I hope HTE will usher in, is a period of science where we can really kind of break genuinely new ground in terms of chemical reactivity, but in ways that we would never have thought of in the past.
So I'm going to name drop my fabulous colleague, Melody Christensen, who's an associate principal chemist at Merck. And she taught me HTE back in the day. And I mean, right now she's working on, using machine learning to optimize reactions. And it's like, she sets them up and they learn, like the algorithm learns from the night before, and it starts to set up the next round. And, after a few days, you have a fully optimized, set of conditions. And that just kind of blew my mind. And I'm just really proud of all of the work that she's done within that area and continues to do for the field of HTE.
I feel compelled to profess my love to Melody Christensen too. Anyways, hey Mela. The thing that seemed kind of science fiction-y to me was the community that has evolved, and I feel like the fact that that we're having this conversation today, I think, is excellent, and I think over the past year, the interest among organic chemists in data science, maybe because the field was reaching a tipping point, or maybe because we spent a lot more time on our computers last year. I'm intrigued by that. I think it was Matthew talking about the throughput and that to get 2496 is definitely something that I think we're very excited about, and I think that's just start thinking about chemistry in that way, because it's just fun, and you can accelerate your research. But even we talk about 1500 as like a bar, right? But then in biochemistry, it's very typical to run 1 million reactions, right? Biochemical reactions. They would typically run 1 million reactions in a high throughput screening campaign. And we're, I don't know if we'll see that in 10 years, really, that maybe we will. I think we're the infrastructure is coming there. But to me, that still remains a science fiction goal, and totally achievable, but hasn't yet been done. I think exciting to think that it will one day soon.
Well, the future might be zero-shot learning, right? We have, physics-based models that can predict the outcome of a reaction without running a single experiment.
I mean, also, what about HTE as art? Like what's in Tim's background? Science fiction.
Yeah.
I thought it was shit.
But I think to your point, Jake, on zero-shot learning, to me, a chemical reaction is incredibly complex. Yep. And I think that I started my career as a medicinal chemist thinking that medicines were super complex. And they are, right? That you have to kind of be soluble and metabolically stable and, selective to the enzyme that you want. But I mean, a chemical reaction, I think requires solubility. It requires stability of the product to all the conditions that it's confronted with. It requires selectivity and, on-target affinities to the catalyst and all that. So I really think that we do need a significantly larger amount of data available before we can really predict every reaction that we want.
Yeah, I 1,000% agree. It doesn't exist. It has to be made.
I think with that, thank you so much for all your thoughts. That's all the time we have for today. For our audience, on behalf of Chem, iScience, ResearchArc, and the wider Cell Press, we hope that you found this discussion as thought-provoking as we did and hopefully inspiring too. And you can take these ideas and implement them in your own lab or your own research program or whatever you're trying to just set up your experiments in the future. Thank you again to all of our panelists for making time in your busy schedule to participate in this discussion. And I hope it was fun too.